So it's with great pleasure that I'd love to welcome Professor Elva Robinson from the University of York to talk to us today about her research into the shining guest ant. So over to you, Elva.
Thank you, and thanks very much for the invitation to talk today about this really quite delightful ant species. It's a species that most people will have never seen unless you go out specifically looking for it. And I love taking people into the field to show them this ant. But since I can't take you into the field today, I'm hoping that instead I'll be able to introduce this fascinating species to you, tell you a bit about it, including some brand new insights, and perhaps inspire you to go and have a look for it for yourselves. So, the theme of this talk is one particular study that I've done recently on the shining guest ant. But my research more broadly looks at questions about how social animals affect their environment and how the environment affects social behaviour. And I do that mostly using ants as my model system, but I use lots of different species of ants in the field and in the lab and through modelling. And I try to answer lots of different types of questions, ranging from quite pure science questions about evolution and behaviour through to quite applied ecology questions around habitat restoration and species translocation. And one of the groups of ants on that I work on the most are the wood ants, and specifically the northern hairy wood ant Formica lugubris, which is in the pictures here. And these species are interesting to me for lots of reasons. And one of the reasons is the huge impact they have on their woodland habitat, so much so that they're termed ecosystem engineers. When we think of ecosystem engineers, we often think about larger charismatic animals like beavers. But ants can also shape and change their environment and create new niches for other organisms. So in the picture on the left here, you can see a wood ant nest in a plantation woodland. Plantations are not really very biodiverse habitats, but when you have wood ants there, you don't just increase the biodiversity by one, adding that single wood ant species. There are also many other species that benefit from the wood ant's presence. The wood ants collect food from a wide area, so they make nutrient hotspots in their nest. And this changes the soil properties in their nests compared to the surrounding area. So we get different soil microorganisms, different soil invertebrates living in the soil of the mounds compared to the surrounding areas. And we have different plants growing on the edges of the mounds too. So overall, if we have wood ants, we have increased heterogeneity and an increase in biodiversity. But the thatched part of the nest above the soil also provides direct benefits to biodiversity by providing living space for other species. So if we ask the question, what is in a wood ant nest, then the answer is hundreds of thousands of wood ants, which if you see them on a sunny day, will all be swarming around on the surface of the nest, as you can see in this video here. These ants are voracious hunters who will eat pretty much any invertebrate that they come across. And if you get too close to the surface of one of these nests, you'll be able to smell the formic acid that they spray for defense and when they're attacking their prey. And you can even see little droplets firing up into the air. So it would seem that a nest is a very hostile environment for anything which isn't itself a wood ant. But despite the hostility of this environment, there are actually species that specialise on living in wood ants. And one of the fun things that I do sometimes is stick my hands in wood ant nests to take samples and look for the different invertebrates which are living inside them. And I should note that I only do this with wood ant nests that can definitely withstand the disturbance and not to too many nests at one site. But if you do go poking around in ant nests, there are all sorts of amazing creatures that live in there that specialise on living in that apparently really hostile environment. And these species include pseudoscorpions, springtails, beetle larvae, mites, woodlice, adult beetles. In total, there are over 100 different species of invertebrate that you can find in wood ant nests. And one of these will be the focus of today's talk, which is the species indicated here with the red arrow. So under the blue arrow here, we have a wood ant, and then there's another really tiny ant. You can see how small it is compared to the wood ant. And this ant is the shining guest ant, Formicoxenus nitidulus. So these two pictures here on the left, again, show both this little shining guest ant and that's much larger host, the wood ant. They're not very closely related species. And as well as you can see how much smaller it is, you can also see how much shinier that little guest ant is compared to the quite matte surface of its host. So that's where it gets its name, the shining guest ant. And on the right here, you can see a guest ant on the surface of the wood ant nest, and it's really dwarfed by the fragments of nest material, the pine needles and bits of leaf litter that form the nest that it's living in. So I mentioned that wood ants are voracious predators. So how does this tiny little guest ant and all those other species avoid becoming ant lunch? So many of the animals that live in wood ant nests have got highly specialized strategies to avoid being eaten. And this can involve, excuse me, this can involve chemical camouflage, so mimicking the scent of wood ants so that the ants are fooled into treating them as another ant. Or it can involve chemical insignificance, so managing to sort of disguise yourself as just a piece of debris in the nest and not having any distinctive smell that gives you away. Or it can even be by bribery, so some beetles and caterpillars produce a tasty gift to give to the ants to placate them. But what the shining guest ant does is actually none of those strategies. It does something a little bit different, and we can just catch a glimpse of that in this video. So it's quite hard to video shining guest ants because they're so small, and when they're on the surface of the nest, they're quite hard to spot. But this is a stone on the edge of a wood ant nest in the North York Moors, and you can see a shining guest ant moving across there, and the wood ant there just walked straight over her. She didn't notice. So sometimes that happens, but sometimes they do notice, which is about to happen with these two down here. Now here comes the ant in, and then there was a little reaction there. She sort of pounced and then kept going. So I just move it back so you can see again. Little pounce. Just one more time so you can see the ant coming in. She's coming towards those two shining guest ants, and then little pounce, and then she drops them and keeps going. So she has detected the shining guest ants. She does that little pounce, but then nothing happens. She doesn't attack, she doesn't eat them. And we know why this is because of some work done by Stephen Martin at the University of Sheffield a few years ago, where he showed that these shining guest ants have on their surface a chemical which tastes really bad to wood ants. So what this is a form of what's called generalized chemical deterrent. So basically it's a bad taste. And in these amazing pictures here by Dan Brown, you can actually see the moment of a wood ant recoiling at that taste. So the wood ant on the left here is actually touching the shining guest ant with her antenna. And if she detected a tasty morsel there, she would immediately pounce and grab that in her jaws. But instead, in the right-hand picture, we can see that she's retreated back, recoiling from that unpleasant taste that she's picked up on the surface of that shining guest ant. So, how can you survive as an uninvited guest? Well, being small and inconspicuous helps because sometimes the wood ants simply don't seem to notice the shining guest ants. But if you do get noticed, a good trick is to taste really bad. So I'm not saying that when you're making your Christmas visiting plans, this is something you should be taking on board as your own strategy, but it does seem to work very well for shining guest ants. And one of the advantages of going for this strategy is that it is a general strategy. So shining guest ants taste bad to all wood ants. So if you can mimic the smell of a species, you can only live with that particular species. But if you just taste bad, you can live in the nests of many different wood ant species. And that's what shining guest ants do. And these shining guest ants, well, they are uninvited guests, but they're not harmful in any way to the wood ants. So there's something analogous to house mice. They live in the nooks and crannies of the nest, inside hollow twigs, and they will steal little scraps of food from the wood ants. But they're not parasitic. They're just taking advantage of this nice, well-protected, stable living space. So let's have a look at who's who in a shining guest ant colony. A wood ant nest will have hundreds of thousands of workers in it, but a shining guest ant colony will only have up to about 100, often many fewer than that. Their whole colony is hidden away inside a single hollow twig somewhere in the thatch of the wood ant nest. And that twig is too narrow for wood ants to get into it, so the wood ants can't find and eat the larvae and eggs of the shining guest ants. So workers, as you probably know, as in all ants, they're female, but they're non-reproductive. And then there's a single reproductive female who's the queen. So she's a bit larger than the workers, and when she's newly emerged, she's got these beautiful wings. But after she's mated, she will lose those wings and spend the rest of her life safely inside the wood ant nest, laying eggs inside a tiny twig. And in the case of shining guest ants, there's just one egg-laying queen per guest ant colony. And then the third adult member of a shining guest ant colony is the male. And this is where shining guest ants are a little bit surprising because, as you will note, this male looks very similar to the worker. And that's unusual in ants. So for those of you who know about ants, you'll know that typically the males are winged. When we get those big swarms of flying ants in the summer, those are winged new queens and winged males all flying around, mating, ready to start new colonies. And that's the norm for ants. The males have wings and they're important for dispersing. But that's not the case in shining guest ants. Males never have wings. And it's quite hard to tell the difference at a glance between this male and a worker of a shining guest ant. But there is one difference which you can see on this picture, is that the male has those 3 little dots on its head there. And those are ocelli, which are rudimentary eyes in addition to the main eyes. And workers in this species don't have those. So that's one way you can tell them apart. The males are produced in the autumn. So during most of the year, the colony is just the workers and the queen laying eggs. And then in the autumn, new queens and males are produced. And the males can be seen walking around on the surface of the wood ant nest. And because the males can't fly off to mate, mating actually happens right on the surface of the wood ant nest. So there's a little video clip here of what that looks like. So in the middle there's a winged queen, a female, and then there are 4 males all clustered around her, and they're all trying to be the one that gets to mate with her. So like all ants, shining guest ant queens can store enough sperm during mating to last them their whole life. So she only needs to mate during this one mating event, and then she won't ever need to mate again. So it's all or nothing for those males, and you can see they're taking that pretty seriously. And there's a wood ant. She just touched them with her antennae and she walked away. She's just not interested in eating them, even though they're pretty distracted right now. So there's 2 more males coming in there. They're going to join in as well. So this is right on the surface of the wood ant nest where there's wood ants wandering around, carrying out their business all the time. There's another male coming in there. And I think there's just, there'll be another wood ant worker is going to come along. There we are again. So she just touched them again with her antennae and then straight away went away. So I think that's probably enough of watching an orgy for today. So we'll move on to where guest ants occur. So shining guest ants occur across Europe. And this is the current data that we have about their distribution within the UK, as collated by the Bees, Wasps and Ants Recording Society. So you can see that they're found quite far north in Scotland, right down on the Welsh borders and down to the south of England. And the distribution of shining guest ants is, of course, influenced by the distribution of their wood ant hosts. In the mainland UK, we have 3 species of wood ant, and these are Formica aquilonia, also sometimes called the Scottish wood ant, which is the teal one up here in Scotland. We have Formica lugubris, the northern hairy wood ant in the picture here, which is in the north of England and also in Scotland. And then we have Formica rufa, sometimes called the red wood ant or the southern red wood ant, which occurs in much of England and Wales. And you can see that the little green squares here, which are the shining guest ant records, occur in areas that are covered by all 3 species of wood ant. But also, as you can see, the shining guest ant distribution is quite sparse and patchy. So it's true that these ants are not very conspicuous, and in some cases, they may be a little underrecorded. but they are totally reliant on wood ants, so they also face some particular challenges when they're dispersing. As I showed you, the male shining guest ant has no wings, so he's not going to do the dispersing. The shining guest ant queen is the only one with wings. So if she's going to fly off and start a brand new colony somewhere else at a new site, then first of all, she has to find a woodland. And that can be quite difficult because in the UK, woodlands are very fragmented, particularly in England. Many pockets of woodland are really isolated. So a queen flying out from a woodland might easily end up in farmland or other unsuitable habitat. So her first challenge is to find a woodland, and then it has to be a woodland with wood ants. Most woodlands in the UK don't have wood ants. Even out of the woodlands which are suitable for wood ants, less than half of them have wood ants in them. Then, even if she is lucky enough to find a woodland that contains wood ants nests are often not very evenly distributed in woods. They're clustered in particular areas. So if she finds the wood with the wood ants, she still has to find a wood ant nest, and then she has to get safely into that nest and set up house. So that nasty tasting chemical deterrent does seem to work pretty well most of the time, but perhaps their wings don't taste quite so bad because this wood ant worker does seem to be attacking the queen by her wing there. They've also got to get into the nest, find a suitable twig to lay eggs in that hasn't already been occupied by another shining guest ant colony that won't share. So a shining guest ant queen has a whole series of challenges if she's going to successfully disperse to a new area. But of course, she could just sidestep some of these challenges. She doesn't have to leave the woodland she's in to go to a new woodland. She could just stay local. She doesn't even have to leave the nest she's in. She could just set up a new colony in the same host nest as her mother. But of course, the risks of not dispersing is an increased chance of inbreeding and loss of local genetic diversity. If she stays on the same nest, she'll have to mate with her brothers, and she'll be competing with her mother and her sisters for space. So one of the motivations for the study that we did is thinking about the dispersal of these tiny little ants. So we have no observational data on dispersal of these tiny queens. Tagging them or following them as they disperse is currently beyond our capabilities. And nobody had done any population genetics on this species either before when we did this study. So there are many unknowns. We know that we've got really patchy populations, but are they completely isolated, or is the potential for gene flow between them? And are those populations healthy? Is the genetic diversity in those populations? Or does each population just descend from a single queen that arrived there and it's only her descendants in that patch? And do the queens even disperse? Or do they just stay very local among the nests really nearby, or even just in the same nest? So these are all things that we really knew nothing about at all, and we still have plenty of unknowns about all of these questions, but I will be able to give you some insights today. The work that I'm going to talk about today was a team effort, so I'm just going to take a moment to introduce my collaborators. Jenny Stocken and I brought an ecological perspective to this work. Joan Cottrell and Stuart O'Hara brought the genetic expertise, and Sandy MacDonald brought the bioinformatics perspective. So I acknowledge all of their contributions. For our study, we sampled shining guest ants from 7 sites in the UK, and these were distributed to cover all 3 UK host species. So up in Scotland, we had 1 Formica aquilonia population, the teal circle here, and 2 mixed populations. And in England, we had 3 Formica lugubris populations and 1 Formica rufa. And at each nest we visited, we took between 1 and 4 shining guest ant males off the surface of the nest. We sampled males to be minimally disruptive to the species because when we did this sampling, we had no information about how fragile the populations we were studying might be, and we didn't want to do anything that would damage them. So we didn't want to take the shining guest ant queens, we didn't want to break nests open and take workers, but the males are on the surface of the nest. They're very numerous in the autumn, and as you saw in the video, there are more males than are needed to keep the population going. So we ended up with 95 samples, and we used these for 2 sets of genetic analyses. So we used the CO1 region of mitochondrial DNA because this is variable on quite a large scale. So it enables us to look at large-scale relationships between populations. And we also used 8 new microsatellite loci, because with this technique, it's possible to look at variation at a finer scale. So in our study, we had questions across 3 scales. At the largest scale, we wanted to look at whether there was differentiation between populations. So can we see genetic differences between the sites that we studied, and how pronounced are those differences? We also wanted to look within a site. Can we see evidence of mixing between the host nests within a site at a local scale? And then at the very small scale, we also looked inside the wood ant nests to find out what we can see in terms of diversity within a single host nest at that very fine scale. And I'll go through the results we found for each one of these in turn. So starting off with differentiation at a large scale. First of all, the mitochondrial DNA results. So we found 6 different haplotypes of CO1 for the shining guest ants in the UK, which are marked as different colours in the key here. So a haplotype, for those of you who aren't into mitochondrial DNA, we can think of that as a variant of this particular mitochondrial gene. And the fact that we found 6 haplotypes is in itself already something useful, because sometimes when you look at this CO1 mitochondrial DNA marker in ants, you find no variation at all at this kind of scale. So finding 6 variants means that CO1 is a useful marker for this species. Each one of these pie charts is the results for one of our 7 sites. So we found that we had one very common haplotype, which was found at almost all of the sites. That's this greenish haplotype 1. But we also had some haplotypes which were found only in England, like haplotype 2, and some which were found only in Scotland, like haplotype 3. So we already had some signs of differentiation between the Scottish and English populations, just looking at the mitochondrial DNA. And we also have signs of genetic diversity within 5 of the sites, even just at this very coarse scale of only having 6 different variants. We can also look at where these UK haplotypes sit within the European context. So other people across Europe have uploaded sequences of this same mitochondrial gene. And in this picture here, the blue dots are the UK samples. So the colour scheme doesn't carry over. All of our UK samples are blue here. And this network diagram is showing how similar the different variants of the gene are, so how much they differ from each other. So UK1 here differs from UK2 just by 1 base pair. So we've got a cluster here of our UK haplotypes, but interestingly, one of our UK ones is also the same haplotype as an ant that turned up in Switzerland. And another one of our UK ones is the same as a shining guest ant that was found in Spain. So this is really interesting and intriguing. It tentatively suggests that the variation we're seeing within the UK captures quite a lot of the variation on a European scale from Spain to Switzerland. So it isn't likely, for example, that all our UK shining guest ants are all descended from the same single colonisation event of the UK. But it also indicates that we need a lot more data, because we had 95 samples and we found 6 different haplotypes from the UK, but the other countries are represented by just 1 to 3 individuals. So we just don't have the same resolution from other parts of Europe to compare with our UK data. But we're working on that. So I'm part of a big European project at the moment, which is collecting data on shining guest ants across the whole of Europe. So we should have a much better haplotype network in a few years to understand where our UK shining guest ants came from, what were their post-glacial colonisation pathways, and how do they relate to those European populations. So that's the mitochondrial DNA at quite a coarse scale, but we also looked at the microsatellite DNA, and here we have a lot more variation. So there's a lot more to work with, and we can see in more detail how our samples cluster. This graph here brings together all of the microsatellite DNA information onto an ordination plot. So each one of these circles or triangles is one of our samples, one shining guest ant, and it looks at how similar they are to each other. And they're coloured by the site which they came from. Those site colours are the same as on the map. The white one wasn't included in this analysis because we didn't have enough data for that site for this analysis. But what we can see straight away is that we have a division between, on the left of the y-axis, these 3 sites here are the 3 sites in Scotland, and on the right side, these 3 sites are the 3 in England. And among these, BX and CF, which are 2 sites in the North York Moors, are clustering very closely together, whereas LS, which is down in Derbyshire, down here, is clustering a bit further apart. So we can see quite a strong geographic signature here. Again, we can see differentiation between the Scottish and the English populations. But we're also not seeing complete genetic isolation. So we're not seeing that each site cluster is completely separate from all of the others. So there is genetic variation within a site, but there's also not complete differentiation between sites, which may indicate that they're not completely isolated in terms of gene flow, or at least that they haven't historically been completely isolated. Within the figure here, we've got samples taken from 2 different host species, Formica aquilonia and Formica lugubris. So aquilonia are the circles and lugubris are the triangles. And we can see that we don't have separation by host species here, because there are some circles here which are mixed in with the triangles. So that means that we're not seeing that all the shining guest ants from Formica aquilonia host nests are genetically different from the ones that we collected from Formica lugubris host nests. We've got no genetic differentiation on the basis of host species. So that's a little bit about genetic differentiation at the large scale. But what about what happens when we look within a site? What can we tell about the relationships between individual shining guest ants from different host nests? So here we're interested in when females disperse, where do they go? What are the patterns of mixing at a local scale? So one possibility is that we get very local dispersal. We know that's the case with the males because they can't fly. So they're going to stay on the nest that they hatched in, or they might possibly walk to a nest that was very nearby, but they're not going to go long distances because, these are 2 millimeter long ants in a woodland. So it's a pretty challenging environment for them. But what about the females? Well, they could also just disperse locally. And if that were the case, when we looked at the genetics at a local scale, we would see something like this, where nests which are very close together are quite similar to each other because queens have dispersed from one to the other, whereas nests which are further away would be genetically more different. In contrast, if females are actually managing to disperse long distance across a site to different nests, we might expect that the patterns of relatedness will not be shaped by distance. So it might be that ants in this nest and this nest are very closely related to each other, even though they're far apart, because one of the queens dispersed from this nest to this nest. So what we can do is look at our data and say, does it follow a pattern more like this or a pattern more like this? So if you're not used to looking at this kind of graph, don't worry, I'll talk you through it. So this is just one example of one of our sites, Feshy Bridge in Scotland. And at this site, we sampled 7 wood ant nests. So these 7 nests, these are the nest codes on the axes here, and we're comparing each nest with each other nest. And we're looking at the genetic distance between the pair of nests, so how different they are. So when we have very low values like 0 or 0.02, which are in dark purple, that means that the ants from those nests are very genetically similar. So for all of these nests, 7, 4, 5, 1, and 8, they're highly similar to each other. When we follow across, follow down, they're highly similar to each other. So that's all of these nests on the map. And this is the scale of the map. So you can see that these nests are over 100 meters apart, but they're highly genetically similar to each other. Now, if all of the nests at the whole site were highly similar to each other, that might indicate an overall lack of genetic diversity at the site, probably due to inbreeding, and then it wouldn't tell us anything about movement patterns. But that's not the case here, because the ants from these other 2 nests are quite different. We've got quite high values here for genetic difference, shown by these paler colours. So this is this nest down here, and also FB6 up here. So FB6, the nest, is right next to FB7. It's really physically close to it, but genetically, they're very different. Whereas FB7 is genetically very similar to FB1, which is all the way over here, more than 100 meters away. So in this example, this shows us that nests near each other are not always the most closely related. So I've just picked one site to talk through here as an example, but we do see a similar pattern when we look across the other sites where we had lots of nests. So before I show you that data, I'm just going to sort of zoom back out again to think about differences between the populations at the whole UK scale. So at the whole UK scale, I showed you some data at that scale earlier, and we could see that if they're closer together in space, they have a lower genetic distance, so they're more similar. If they're further away in space, they're more genetically different. That's between populations at the whole UK scale. The question here is, if we look within populations, do we see the same relationship? And if there's local dispersal only, if queens are only moving to nests nearby, we would expect the same kind of relationship where when nests are very close together, we've got low genetic distance. Notice we're on a much smaller scale on the kilometers here because this is within populations. So now for our actual data, and what we see is no relationship at all. We don't see this positive relationship here. And that also holds statistically. So here we have a statistically significant positive relationship between genetic and spatial difference. But when we look within populations, we don't have that relationship. So we've got some nests which are very close together that are highly genetically different. And we've got other nests which are very far apart physically, but really genetically similar. So overall, what this means is that we're not seeing evidence for this top picture here, where we've got local dispersal only. Instead, what we're seeing is evidence that fits more like a pattern like this, where we've got some events of long distance dispersal within the site. So we've got good evidence of mixing within sites at a local scale, and we've got genetic diversity at those local scales too. So what about this really fine scale? What about what's happening actually within a single wood ant nest? So we know that wood ant nests can have several shining guest ant colonies inside them. We know this because in the past, people, not me, have actually taken a whole wood ant colony apart and they've counted all of the shining guest ants. And they found that there are multiple shining guest ant colonies in separate twigs, each with their own queen inside a wood ant nest. But that could mean, as I mentioned earlier, that the queens are just choosing not to disperse. So when queens hatch within a nest, even though they've got wings and they could fly, they could also choose not to and just stay in the same host nest and make a new colony in that nest. That reduces the risk of failing to find a new host nest, but it increases the risk you'll end up mating with your own brothers. If that happened, we would expect to see something like I've got over here on the left, where all of the male ants that we pull off the surface of this wood ant nest come from the same genetic matriline. So they might not come from the same colony, but the queens of their different colonies would be highly related. So they'd be sisters, or one is the daughter or granddaughter of another. And then in a different host nest, we might find that this nest has a different matriline because it was a different queen that arrived there originally, and all the ants that are there now are descendants of that same queen. In contrast, if queens actually move to new nests and establish their colonies away from their home nest, we would expect to find a mix of genetic lines within that host nest, as we have over here on the right. Now, of course, in our study, we've made this a little bit difficult for ourselves because we're only sampling 1 to 4 males. Our chances of detecting multiple matrilines, even when they're present, is not 100%. So in the case of this nest here, if we sample 2 ants and they happen to both be the blue ants, it would look like just one matriline. But if we sampled 1 red and 1 blue, we would know that we did have 2 matrilines. So when we look at our data here, overall, we found that in 47% of the nests we looked at, we found evidence of multiple genetically distinct queens. This is combining data from both the mitochondria and the microsatellites. So in 47% of the nests, which includes examples at almost all of the sites we looked at, these black bars are the counts where we had evidence for multiple genetically distinct queens. In the other 63%, 53%, sorry, the other 53%, we didn't have any evidence of multiple queens in there. So all the males we looked at likely came from the same matriline, but that doesn't mean for sure that there weren't multiple queens in that host nest, because we only sampled 1 to 4 males. So there could still have been multiple queens in there. So that 47% is a minimum baseline for the proportion of host nests which have multiple queens. The true number could be much higher. So we've looked across these 3 levels, the large scale, the local scale, and the fine scale. And what have we found out about shining guest ants? Well, at that large scale, we found that the Scottish and English populations are genetically differentiated from each other. They're different. We find that there is within site genetic diversity. So all of the ants at one site, they're not highly inbred, and they're not all descended from a single individual. There is within site genetic diversity. And those populations do not show complete isolation from each other, so it could be that there is still ongoing gene flow. We find that the CO1 region of mitochondria shows enough variation to be useful for studies at a European level to find out about the post-glacial colonization history of this species and how that relates to the movement of their wood ant hosts. At the more local scale, we don't find any evidence for host specificity, but we only had enough data to look at that from one mixed host site. So there's maybe more work to be done there. But we didn't really expect host specificity either, because these ants use that generalized chemical deterrence strategy, which is not really tailored to specific host species. We do find evidence of effective mixing within sites, suggesting that at least some queens are dispersing within sites quite successfully. And then at the finest scale, when we look within a host nest, almost half of the host nests had multiple genetically distinct shining guest ant colonies in them. So again, this indicates effective within-site dispersal because queens are successfully establishing colonies in new host nests. But this also has some conservation implications, because it suggests that any individual wood ant nest that is occupied by shining guest ants is a valuable conservation unit, because it's potentially providing a habitat patch for many different shining guest ant colonies. At some wood ant sites, most of the wood ant nests have shining guest ants in them, but that's not the norm. Quite often, shining guest ants are actually only in a few of the wood ant nests, and the majority of the wood ant nests have no shining guest ants in them at all. We don't really know why they seem to prefer some nests to others, but it seems that at some sites, a small number of wood ant nests could house a high proportion of the shining guest ant genetic diversity, so we really need to take special care of those wood ant nests. So, if I've convinced you that these ants are interesting and you want to go out and see them for yourself, how can you find these beautiful ants? Well, I have a 3-step plan for you for how to go and find these ants. So, step 1 is first find your wood ants. Okay, that's the very first thing you have to do, because you won't find shining guest ants if you don't first find wood ants. If you don't know where to find your nearest wood ant population, you can go and have a look on the BWAS website, the Bees, Wasps and Ants Recording Society. They have maps of the different wood ant species, so you can find out where your nearest wood ant populations are. Fortunately, wood ant nests are very conspicuous, so if you take a walk around the woods in an area where there's been a recent wood ant record, you shouldn't have any trouble finding them. So once you've found your wood ant population, you need to pick the right day. And it's a little unfortunate timing that I'm giving this talk in December, because now you have to wait a little while before you go and do this. But I hope you won't forget how beautiful and special these tiny ants are when it comes around to late summer next year. So exactly when is good to go depends a bit on how far north you are. In the south of England, the window is longer, from July onwards. In Scotland, you might not want to start looking before late August, and it needs to be the right weather. So wood ants are still active on the surface of wood ant nests. Sorry, shining guest ants are still active on the surface of wood ant nests right through to October. when it's a sunny day. The best weather to see them is sunny. It is possible to see them when it's overcast, but they're more numerous when the sun is out. But not if it's actually so hot that the wood ants themselves are hiding. If it's too hot for the wood ants, then it will also be too hot for the shining guest ants. And if it's so cold the wood ants are also hiding, it's too cold for the shining guest ants too. So you want a day when the wood ants are active on the nests. And then all you have to do is watch the wood ant nest surface. And we've done some experiments with this of watching for various lengths of time. And I would say if you wait 3 minutes and you haven't seen them, you're probably not going to see them. So try another nest. And yes, if you lean over a wood ant nest for 3 minutes, the wood ants might start to get a little bit annoyed with you. So I would recommend you tuck your trousers into your socks, and it's probably a good idea to wear glasses or sunglasses. I don't wear glasses myself, but I do wear sunglasses when I'm doing this so that wood ants don't squirt me in the eye with their formic acid. But wood ants are usually quite calm in the autumn when the weather is cool, and it's definitely worth putting up with a few annoyed wood ants to get that amazing moment when you see the tiny shining guest ant running across the nest. So if you do see some guest ants, it would be great if you could submit records through iNaturalist or iRecord or directly to BWAS. But if you don't spot any shining guest ants, you can still submit the record for the wood ant nest through whatever usual route you might use. But also we have a European-wide citizen science project running at the moment for the next couple of years, where we're systematically collecting wood ant data across the whole of Europe. So for this project, we don't just want to know about the locations of nests, we're collecting data about nest size and the habitats around the nest. So it's just a few additional questions that are very quick. So if you would like to do this, if you're going to go and look for shining guest ants, it would be fantastic if you could take a look at the Monitant website, follow the citizen science link, and you can submit the records there through our iNaturalist project, or if you don't use iNaturalist, we've got our own toolbox. It's really quick and easy to submit the record there. So it would be fantastic to have some more UK wood ant data contributing to this rich data set from across Europe. So finally, I would like to thank the collaborators on this project, the co-authors and other people who contributed to the planning and data collection, all the people who created and shared amazing images and videos. I'd like to thank our funders. The core of this work was funded by Future Woodlands Scotland, and all of the site managers who gave us access. And thank you very much for the invitation to give the talk today.